So the fact that these terms exist implies that there's a specificity to the vesicular trafficking process such that this particular vesicle is going to fuse with the Golgi and not with the lysosome or the plasma membrane or any of these other vesicular systems. And that implies that the process, even as early as the budding that produces this vesicle, already has the information to specify where it's going to fuse. And that implies that the cargo, which is ultimately signifying the direction of vesicular trafficking, has the ability to communicate that information, even though it might be a soluble luminal component with no parts exposed to the cytoplasm, and yet it's determining how that process is going to work indirectly. So what we'll do is talk about the specifics of the budding, translocation, and fusion reactions or mechanisms in a generic sense, because that will allow us to uncover the mechanisms that account for that specificity. So we'll do that for quite a while, and then we'll go back to talking about some of these particular trafficking pathways. So we're going to go ahead and get that cargo out because we've gone this far. So we'll talk first about anterograde trafficking as the first example of a specific trafficking mechanism. It is the one with the least number of components. So although it certainly isn't fair to say that it's simple, it's at least got the fewest moving parts. And that's good in a way. It's bad in another sense that it doesn't, because of the few parts involved, doesn't exactly allow us to talk about all of the concepts associated with vesicular trafficking. But we'll talk about that, and then we'll get the cargo out here somewhere. We're going to go very quickly through exocytosis. It's not particularly well known, for one thing, and we'll talk about exocytosis in a different context, that is neurotransmitter release, when we talk about nerves. So we'll go through that quickly. We'll talk about retrograde trafficking only to the point of talking about why it exists, because it's not intuitively obvious based on what we've done so far why we need it. So we'll talk about that, and also to introduce this compartment, the so-called ergic, ER Golgi intermediate compartment, because I want to talk about coronavirus, COVID-19 in particular, and what this kind of translocation and that compartment has to do with it. Basically, this is the factory for the production of the infectious virions. So we'll talk about that a little bit. And then wrap up the discussion with endocytosis, which is by far the longest known, much more complicated than the others, but it's a really good segue into the next big topic, which is signaling. So we'll finish up there. We'll do a brief detour to the lysosome and use that as a reason to talk about autophagy, which is a concept that's really kind of hard to exactly fit anywhere, but while we're talking about all these membrane fusions, it's a reasonable place to talk about that. So the overall idea is to account for most of the mechanisms associated with the specificity and the physical movement of these things, and then wrap up with something that is also going to be involved in the ability to communicate with other cells. So we're getting down to the business of kind of a generic trafficking reaction. This is an early figure in chapter 13 of your text. And this figure is really designed to focus on the cargo, so they didn't even actually talk about coats, but I'm going to. But for one thing, we have a convention as far as the cargo. Soluble luminal cargo is a red dot. Integral membrane proteins that are cargo are green in this diagram. So the overall point here is that there is a donor membrane, which we can keep generic, and a target membrane that we can keep generic. And if we do that, then the overall process is designed to get the soluble cargo into the lumen of the target membrane and get the transmembrane cargo into the membrane of that target system. So the overall process begins, as we mentioned earlier, with budding, and I think this is the appropriate place to talk about a principal component in the budding process, and that is the coat. This is one of the things that distinguishes the individual trafficking reactions, although it doesn't determine the specificity of the process. So it's not going to answer that question, but it is specific for processes, and it is an instrumental part in the budding and the communication between the cargo and the overall process, as it turns out. So this thing in purple generically would be a coat, but the coat content is going to depend on which type of reaction, which vesicular trafficking direction we're talking about. So before too long, we're not going to talk about anterograde anymore, and we're going to call it a COP2 reaction because this is the kind of coat that's associated with that direction. So we'll describe the composition of the things that are collectively referred to as COP2. This is COP protein complex is what that stands for. Complex 2 is the one that goes that is involved if this donor membrane is the ER and this is the Golgi, then this would be COP2. COP1 is the coat associated with retrograde transport. So if this happened to be the Golgi and this the ER, the backwards direction, then this thing would be COP1. And then if it's anything from the trans-Golgi network out to the plasma membrane or back again, then chances are the coat would be clathrin, by far the longest known coat protein of them all, and the one that's involved in the biggest variety of reactions. So this isn't to say that this is all the coats, or the coats are always required for vesicular trafficking, but it's complicated enough, so this is all we're going to talk about in this section.